Hey guys, so today we're going to be talking about DNA replication. And so just what is DNA anyway? So DNA is the building block of all life. It contains the genetic information that is responsible for all parts and functions of your body. But more specifically, DNA is a nucleic acid, which is one of the four major biological macromolecules. And as a nucleic acid, DNA is made up of nucleotides, which is the monomer of nucleic acids. I'll make one for you real quick. So a nucleotide contains a phosphate group, a 5-sided sugar, and a nitrogenous base. And the structure should look something like this. So you have your 5-sided sugar, classic pentagon. This is your sugar, which is attached to your phosphate group up here. This is your phosphate group. Phosphate, oxygen, and last we have our nitrogenous base, which will be up here. And then, okay, so here's our nitrogenous base. So this is a basic nucleotide, and this is what makes up the DNA that makes up all of us. Alright, so the phosphate group and the 5-sided sugars of the nucleotides, should keep this out, are made together, or are stuck together with covalent bonds. So these components are stuck together by covalent bonds. And then the nucleotides, as they connect together in a chain to form DNA, are stuck together by these things called phosphodiester bonds. And then the bases, this base will connect with another nucleotide's nitrogenous base. They're bonded together by hydrogen bonds. So the bases, adenine, thymine, cytosine, and guanine, are paired together and arranged to create unique combinations to store genetic information. And so the trait of carrying genetic information is not unique to DNA, as RNA can also store genetic information and replicate itself. But DNA's double-stranded structure provides security and stability for the genetic information, while RNA may damage and deteriorate it. So DNA is used as the hereditary model due to its safety in reproduction. So the adenine and guanine bases are called purines, while the thymine and cytosines are pyrimidines. And so adenine can only pair with thymine, and cytosine can only pair with guanine. And so the sequencing of these bases is what determines what the DNA codes in the body. And so, I don't have a great example here, but DNA strands are structured as a double helix, which means that there are two individual strands of DNA, or two individual strands of nucleotides, which connect together and form sort of like a twisted ladder. So you've probably seen it a lot, this double helix structure. So these are all the nitrogenous bases connecting together with hydrogen bonds. And then these are the two nucleotide like backbones. And so obviously each new cell needs genetic information when it divides. So DNA must also be replicated so that there's enough genetic information to go around between all the newly created cells. So this leads us to DNA replication, which is our main topic today. So how does this single strand of DNA here split up into two new strands of DNA? So first we have our most important components to this replication. We have mostly enzymes here. Helicase, we have primase, DNA polymerase, and ligase. Where's my ligase at? Here. This would be our ligase. So the helicase acts as a zipper pretty much for this single strand here. So it'll attach to the strands and begin to unzip it. So if these are connected right now, it begins to separate the strands. So they open up and look something like this. So it breaks the hydrogen bonds in the DNA and separates the two strands of nucleotides. So the zipper opens it up. Another important thing is that the tension that the helicase puts on the DNA when it opens up normally would cause the DNA to supercoil and like constrict all over itself, and that's obviously not great if you're trying to keep it straight for replication. So topoisomerase is here to prevent it from coiling up. So in addition, we have these single-stranded binding proteins, or SSBPs, which are responsible for keeping these the replication fork, which this is called, from closing back together in replication. So these SSBPs bind along the edges to keep it from closing in on itself. So primase right here produces a primer which attaches to the half DNA strand and produces primase produces a primer, right, which is just an RNA strand, which allows DNA polymerase to know like where to go, where to bind, and where to begin its work in replication. So originally we have half a strand here, and then we add this RNA primase, or in the RNA strand primer, to complete like kind of a complete strand here, so that the DNA polymerase can bind on these primers. All right, so for this next part, it's important to understand that DNA is anti-parallel, so the strands can run from like a 5' to a 3' or a 3' to a 5'. So here we have, this will be the 5' and the 3', and the strands run in opposite directions. So this side will be a 3' to a 5'. So they go in opposite directions, and this will create some problems later on. We'll get to that in a second. So DNA polymerase is responsible for actually replicating the DNA molecules and completing the half DNA strand that we have here. The DNA polymerase enzyme joins together DNTPs, or deoxyribonucleoside triphosphate, which are the building blocks for DNA. So as an enzyme, it just catalyzes that reaction. They all join together, and the new strand is made as the polymerase progresses. So it goes along and completes the base pairs, so that we have an identical DNA strand to the original one on both sides. So at this point, we'll have the workings of two new strands. The DNA polymerase is also a proofreader, so it will identify and correct minor issues in the DNA replication, but the final proofreader is exonuclease. So this exonuclease follows behind the DNA polymerases as they progress, and they clean up any extra errors that the DNA polymerase might have missed. In addition, the DNA polymerase is actually, like, although we just call it DNA polymerase, it's actually separated into 3 different DNA polymerases, DNA polymerase 1 through 3. And so DNA polymerase 1 and 2 are responsible for the cleanup of primers and proofreading the newly written DNA. So it'll clean up any primers that are there and get rid of them. And then the DNA polymerase 3 is what actually like writes the DNA. It's what creates the new DNA. Okay, so this replication occurs on both strands. So we have the primers out of here due to DNA polymerase 1, 2. And we have exonuclease cleaning up, and DNA polymerase is moving along. So now we have 2 copies of the original data on the sides. But DNA polymerase can only move from the 5' end to the 3' end of DNA, which creates some problems because obviously we have a 3' end here, so it'd have to move from left to right instead of right to left, which works against the helicase, as the helicase is moving to open up more of the DNA as it moves along in this replication fork. So we have this DNA polymerase moving along with the helicase. This is called the leading strand, and this DNA polymerase is moving against the flow of the helicase, called the lagging strand. So since the DNA polymerase on the leading strand moves with the helicase, there's very few issues, and it can create DNA continuously as there's more genetic material being opened up by the helicase as it moves along. However, with the lagging strand, since it's moving away from the helicase, the DNA polymerase cannot continuously produce the new DNA. It eventually reaches a dead end right here, and it has to fall off. So the solution to that is simply adding another primase and primer close to the helicase, and simply adding on a new DNA polymerase where we left off to continue the chain. However, it kind of, since it starts and stops, the DNA polymerase falls off and has to start again. You get these sort of chunks of new DNA, and these chunks are called the Okazaki fragments. And so that's what we have our ligase for. So the ligase is like a glue. It goes along and it cleans up the fragments and kind of glues everything together so that we have a continuous strand of DNA. So now each strand of DNA contains one newly replicated strand and one strand from the original piece of DNA. This process is declared semi-conservative because the original content is never destroyed. You always have these original strands along with two new strands that are produced as you go along. So the semi-conservative nature of DNA replication allows DNA to safely pass on genetic information with a very small margin of error. So the original content is never destroyed, so you can safely reproduce with low risk. And so the final issue is that with a leading and lagging strand, there are leftover unpaired bases on the ends. It's not pictured here, but there's, if you can imagine, extra unpaired bases on either end here. And so if the strands remain unpaired, they'll be cut off and the genetic information will be lost, which we don't want. So the solution that the DNA has come up with is telomere replication, where a telomere a telomerase will go along and add just extra information onto the end. It's information that doesn't code for anything, but it allows, the extra length allows the DNA polymerase to reach all the genetic information that does code for something, so that the important genetic information isn't cut off when the strands are separated. So there's as little genetic information loss as possible with DNA replication. So it's a very safe process. Again, so the telomerase it has its own RNA, and it acts as a primer, so it can attach on and kind of act as its own primer to know where it needs to go. Then it just moves on and extends and creates more unpaired bases so that you can sort of keep the genetic information that you need, that is important to the function of the DNA. And so that about sums up DNA replication. Thanks so much for watching.